welcome again to our fall course, the Virtual Brain in Clinical Research. And today I will give you an overview of the entire course and the topics that you will see in detail in the subsequent sessions. So first of all, who we are. Who are we? We are the team at the Berlin Institute of Health at the Charite. So we are located here behind this large Charite patient building. This is the building where we are located under this green roof in the center of Berlin. And in the course of the course, you will meet several of our lab members. As I mentioned, we had many registrations. The course is free and open. Originally, it's a course that we prepare for the Bernstein Center for Computational Neuroscience, and in particular for the master students and for the PhD students of the Charité. But we opened the course during COVID for international participants, and we noted that there was a really big interest. So this time we had registrations from 21 countries, and you see most of the participants are from Germany, but about the other half of the participants is from other sites, Eastern Europe, Africa, Asia, South America, North America. So we are really international, and this is exactly what we would like to see. And this also opens up, of course, opportunities for new collaborations, for getting in touch, and maybe some of the participants here are interested in doing their master thesis or PhD project in the future in our lab. So if you're interested, just feel free to reach out anytime. You find more details about the course on our website, our lab website, and I just put the link again in the chat here. There you find the full agenda, and there you will also find the link for the video recordings of the individual sessions. And this is our program. We have 12 sessions planned. You'll find the titles and the names of the speakers on our website. And you will see that we have many different topics. The focus here is on computational brain network modeling for clinical applications. So the session after this session and the next week will be an introduction to the software, the virtual brain. Yeah, that is the basis for our research. And then you will dive into many different clinical topics. We will speak about neurodegeneration. Dr. Leon Stefanowski, who you saw is present here today, also will present this session. Then we will learn by Leon Martin, who also is here today, how to integrate different sources of knowledge in our brain network models to further refine them and make them biologically more plausible. We will see a session on mechanisms of psychosis and how we investigate them with brain network modeling and other methods. We will see another session how we combine AI and brain network modeling in order to, for example, develop biomarkers that predict disease trajectories or allow for better diagnostic classification. We will see a session on multi-scale co-simulations. This is a development of the past couple of years, where we can combine simulators that simulate neuronal activity at different degrees of detail. And maybe Dionysus also touches on the interface to the neurorobotics platform. That means that the virtual brains can be coupled to simulated robotic devices, and you can see the functional output of the activity. We will see an application for deep brain stimulation optimization presented by Dr. Jill Meyer. You will also learn about the processing pipelines that translate the raw imaging data that we use for personalization of our models into the derivative data that can be directly incorporated in the virtual brains. And specifically, there's a pipeline developed for lesioned brains also that will be shown in more detail. Then there's another novel development, TVB for positron emission tomography, that will be presented by Dr. Holger Taher. And also very recent work, this will be probably the session that will not be recorded, on the principles of traveling waves and new insights in this respect, presented by Dominic Koller. And then last but not least, actually it's even hidden by my other screen, we will talk about the GDPR compliant workflows that are being developed, and also the infrastructures that are being developed by our team to enable digital twin technologies with a proper protection of the sensitive information, biometric information in these models. And this will be presented by Dr. Michael Schirner. So this is our plan. At the end, as mentioned in the description of the course, there's an examination, a written examination, and this is a precondition for getting the ECTS. And how many, whether 2 or 3, this depends actually locally on whether you are in the Charite PhD program or a master's student at the Bernstein Center. But in any case, you get a certificate of your participation and the earned units if you successfully pass the final exam. So, as the name of the course states already, the focus is the virtual brain simulation platform and the applications based on this methodology. You find the virtual brain simulation framework on this website, thevirtualbrain.org. And there you find, for example, also the software that you can download. Later on, I show you how you can use the simulator even without downloading it and installing it locally. But maybe this is a good time to see who has used already TVB and maybe already installed it on your devices. So maybe you could give us a little hint by just showing your hand if you have used already TVB before. Please raise your hand. So I see our own lab members raising their hand. But I don't see much more hands. Maybe this is because I don't see a lot here with my presentation modus. Maybe one of the lab members, Leon, can count the hands. I'm not sure I see them all now, and tell me the number of raised hands. So Leon, can you help me here?
I think it's 7.
Okay, and most
8.
of them probably are from our own lab. Yeah. Okay, then there are many new individuals who are new to TVB. This is very good to know. This means that we really should start from the scratch, yeah, and providing you yeah, an easy entry in this new technology. So thanks again for showing your hands, and you can lower them now again. So this is the main entry point to the tool that we are using, and I recommend going to this website. There you will find a lot of information. So for example, here you see advertised the EduPack. Yes, these are tutorials that we update from time to time, where you have video lectures, often accompanied also with data and other resources that you can use to step-by-step follow the demonstrations and run your own simulations. So this is a very good and easy starting point. Maybe a little bit more advanced, but also very useful is this page. I just put the link again in the chat. This is the entire documentation of the virtual brain software, including links to multiple notebooks, yeah, that provide use cases, how you can operate the virtual brain via the command line interface. So this is also a very recommended resource for you. There's another possibility. You can go to the INCF training space, the International Neuroinformatics Facility, where you also will find a lot of material. It's not completely redundant with what you find on the TVB website. So I recommend also checking out the courses there. They might be complementary. And then last but not least, and I apologize that there are so many resources, this can be already overwhelming, but we will guide you through all of the topics, of course, during the next 12 weeks. But this is our own YouTube channel. And there you will also find the recordings of the past fall and summer schools that we have conducted. So this might be also helpful for you. You see here, for example, the last summer school playlist and many other playlists that could be of interest. Okay, so these are the resources. I'm sure that my colleagues later on will point you again to the relevant resources during their presentations and sessions. But now we start diving into the actual topic, the virtual brain. What is it about? It's about personalized brain network simulation. So we combine data of individual people or subjects, could be also animals, with mathematical models to simulate their brain activity on the computer. This could be the notebook if it is more an abstract coarse-grained model, or if you do large parameter explorations or run more fine-grained simulations, you need probably high-performance computing. And as mentioned, it's about brain network models. Yeah? So the core idea is not to simulate every cell of the brain, but to reduce the brain to brain regions, then use data, imaging data, to infer the connectivity between the regions and construct a brain network model. When each node, as you can see here, we have mathematical equations that describe the population activity of this node. And these population models, or mean field models, or neural mass models, can be of different degrees of detail. Yeah, they can be very simple oscillator models, or they can be more detailed and really describe the state variables, the activities of biological processes, for example, the firing rates of inhibitory groups of neurons or excitatory groups of neurons, of pyramidal cells, of inhibitory or excitatory interneurons, and so forth. But this is the idea, that we reduce the brain and that we are not trying to simulate every process, every cell, every interaction, every molecular process that is even on a subcellular level, because this is not feasible. We have shown that with this approach, meaningful predictions and simulations can be achieved that can be even relevant for patients and for clinical applications. Here you see the main website again, as mentioned, the virtualbrain.org is the URL. And I think I made the screenshot yesterday. So this was the number of downloads at that time. So we are approaching now, since the first release in 2012, 50,000 downloads. This is not the only way to get a copy of TVB, but the way that is most used. So this number is a good estimate of the number of downloads of the software. There are some other resources where the software actually can be retrieved that does not is not considered here in this number. So maybe in the course of this course we will reach the 50,000. I'm quite optimistic about this. We celebrated last year 10 years of the virtual brain, as mentioned. Stop this count. The first release was in 2012, and last year in 2022 at the SFM conference in San Diego, a large group of people, actually. You see only the founders, some core people, but On the other side, not in front of the camera, but behind the camera, there were the wonderful people who actually prepared the cake and the party. And we had a big celebration together. So here's Victor Gierse, one of the co-founders, Randy McIntosh, Jochen Meersmann, Anna Solotkin, and Steve Small, some of the core people behind the virtual brain. So the virtual brain actually was online as open source platform 1 year before the Human Brain Project started. This might be a fact of interest. If you download the software and start it running, you will see this initial page where you also see the core institutions that lead the development of the platform in Marseille, ex-Marseille University, Baycrest, and actually now the person, Randy McIntosh, moved from Baycrest to Vancouver, to the Simon Fraser University and the Charite. And if you want to get a nice introduction to what this whole thing is for, I recommend our early publication, The Virtual Brain Integrates Computational Modeling and Multimodal Neuroimaging, where we describe the idea. And this was published just a couple of months after the first release of the software. And Michael Schoner actually was starting his PhD at that time in my lab, and now he is a senior researcher, and you will see a lot of work of him that he has done in the past 10 years with the virtual brain. So the idea is to use theory and mathematics, mathematical models that describe the complex interactions in the brain and enrich the mathematical models with personal information to individualize our models. And for this, we use multimodal imaging data. And this will be demonstrated based on many use cases during the course. But these are the most typical ingredients. The structural connectomes derived from diffusion tensor imaging data, the cortical surfaces derived from anatomical data, maps or parcellations that are based on anatomical landmarks or other atlases. We also have functional parcellations. There are many different flavors available. Then functional magnetic resonance imaging. This is used to tune usually the model to get it in a regime that is close to the biological regime or the regime of the individual. And we can use for this purpose also electroencephalography data or also MEG data. The addition of PET is relatively new, therefore it's not mentioned here, but it's also a possibility to integrate positron emission tomography data. As mentioned before, we can not only simulate human brains, but also animal brains. This is an example of a Markek brain network model. And we are now collaborating with Magdeburg in a collaborative research center where we advance this stream of research. But you will also see that we can simulate rodent brains as well. So the brains of mice and rats, for example. Oops, just want to move forwards actually. Yeah, this is again the main concept. We have a brain network model. We derive the connectivity between the brain regions from, for example, diffusion tensor imaging data. And then we place in each individual brain region a mean field model or neural mass model. And this is a set of usually differential equations that describe to a more or less detailed degree the activity of neuronal populations, of groups of neuronal populations. This, for example, is a reduced von Wang model that consists of excitatory populations and inhibitory populations that are interacting. And these populations then interact with each other via the structural connectome and influence their mutual dynamics. So normally we are not with TVB simulating single cells, but in the course of the last years, we have developed a co-simulation approach where it is possible to replace some of the mean fields or neural masses by spiking neural networks. And with this, we have multi-scale co-simulations. We use other simulators like Nest, Anarchy, to run the simulations at the more detailed level. And with this, we can see how the large-scale dynamics that are captured on a coarser scale influence the fine-grained dynamics in a spiking neural network, for example. And vice versa. Of course, the spiking neural network also has an output that then goes in the whole brain dynamics and influences the whole brain dynamics. And if there's plasticity, for example, and changes due to plasticity in the spiking neural networks, this might also lead to changes on the global scale then. So this is the simplified large-scale equation of TVB. So we describe the activity as a function of time. We have a population model indicated here by F that consists of a set of state variables. And we compute the activity as a function of time, as a sum of the instantaneous activity of the populations and the weighted sum of all the connected populations or neural masses. And the weighted sum takes into account the weights that come from the structural connectivity matrix. So for example, from the diffusion tensor imaging data and tractography. The weights that characterize the connection between individual region pairs. And we can also take into account the time delays that are a function of the distances between the connected region pairs. This is just a noise term on top of the activity. We can detail this a little bit more. So this is another visualization of the large-scale equation of the virtual brain. We have the differential operator that maps the system of equations into its derivatives. We have the neural mass model, and there we have actually now about 30 different flavors or kinds of neural mass models you can select from in the virtual brain software. We have the set of state variables that characterize the neural mass model. We have a global coupling scaling factor, and this is needed because the structural connectivity usually is obtained from tractography data and do not provide absolute measures of connectivity. So therefore, there needs to be an adjustment that is achieved through a scaling factor. We have the structural connectivity matrix that provides the long-range coupling weights. So the degree of connectivity between the region pairs. We have the before mentioned distance matrix that determines the time delays between the regions. And if you like, you can also add local connectivity and even run surface simulations. I'm not sure whether we demonstrate this in the course of the course, but it's a possibility also to even add more detail to the simulations. Then we have the injected activities. This could be, for example, a stimulation. We will see this during the fall course. And at the end, we have again the noise term. And this is just another illustration of this large-scale equation of TVB, another simplification or structuring that you really understand the idea. This is activity. We have the neural population activity that is provided by the neural mass model. And the activity or the trajectory over time is a function of this neural population activity. The long-range input connected with a specific neural mass and the short-range input coming from the local connectivity. And if you inject activity, if you, for example, simulate stimulation, you have an additional term and a noise factor. So these are the ingredients of a virtual brain as we have it in TVB. So now, as mentioned already, if you construct such a model, usually you will not directly get biologically plausible activity. You have to do some tuning with the parameters, and one of the most important parameters is the global scaling parameter. And you see here that we change this. On this axis, you see the global scaling factor. Here you see it as well. And what we look at here on the left side is the empirical functional connectivity of this example and the empirical functional connectivity dynamics and the simulated functional connectivity and simulated functional connectivity dynamics. Here in the middle, we see EEG activity in our simulation output. And here in this panel, you see how well the simulated and the empirical activity correspond. And we have different measures depending on the empirical data, be it functional connectivity dynamics, or the EEG signature that we are looking here. But interestingly, what is very clear from the simulation, there's only a certain window of the parameter G where for all the three metrics, we get a good correspondence between the simulated activity and the empirical metric. And this is this window. Let us just wait a second until we reach the window again, and then we will see that the simulated functional connectivity dynamics, and the EEG dynamics become biologically plausible. So they correspond to their empirical counterparts. This is now the time. So we need to find the sweet spots of our parameter spaces where we can generate biologically plausible activity. And this is the purpose of the parameter tuning exercise. So now let me just jump back where you can actually get TVB and TVB related resources. You can go to the website of TVB, but you can also go to eBrains. eBrains is the digital infrastructure of the Human Brain Project that started in 2013, a European project. So it started 1 year after the first release of TVB, and TVB now plays a core role in the eBrains platform. So let me just give you the link. Chat. So this is the website where you get to the resources of TVB on the eBrains platform. Why is this important? Because there you have the possibility to even run a simulation in the cloud without installing TVB on your local machine. And I can actually demonstrate you this. Let me just change what I share. And now share my browser. 1 second. Okay, I hope this is the right window. And this is now the same site that I have shown you on the slide, that you can reach via the link. And you can just do the same that I'm doing now in parallel. If you scroll down, you will find the eBrains Cloud Simulator, and you can click it. And I'm already logged in eBrains now. Probably you have to provide your eBrains credentials. And if you are not yet a registered user of eBrains, you have to register for eBrains. Maybe one of the colleagues can give you the link, so you can just search for register eBrains and you will directly find the website. But maybe that we can also provide it in the chat. So if you have registered, you just use your eBrains credentials, and then you directly can start the software that you can also deploy on your computer. You have now the graphical user interface. You see exactly the same information. And you have the full functionality already available, yeah, of the virtual brain. You can, for example, go to data structure. Just move now this control panel of Zoom. And there you will have already many toy data available, which you can use to set up your simulation. So there are really many data, many connectomes. Let me just scroll down. So maybe we can make a visualization here. So if we, for example, choose the surface of this toy data set, we can go to visualizers. select surface visualizer. And there we have the surface visualized using the software in the cloud. And just to give you an idea, so the graphical user interface is really a nice starting point to get familiar with the software. You can have nice visualizations. I just added now the face. It's a little bit difficult to control. And of course, you have the simulator cockpit here and can set up a simulation with the data that are already available, so you don't have to deploy anything on your computer. Okay, so let me go back to my presentation. So there's one publication where we actually summarize all the tools that have been deployed on eBrains that are directly accessible there. You can read the details and also find the links to all the open source repositories where the software and code is available. What you see here looks complex, but what it shows is that the services of TVB are interacting and interfaced with the core services of eBrains and also with other applications that are provided by other developers and research groups. So as I have demonstrated to you, we can, for example, run the virtual brain directly in the cloud. This is an OpenShift environment of eBrains. We have a single sign-on, yeah, so the authentication is unified with the authentication of eBrains. So there are not multiple logins. It's easy for the users. And there's a high-performance computing backend, so you can run compute resource simulations using the supercomputers in the backend provided by this European infrastructure. And there are many more resources, some of which you will see either today or during the presentations of my colleagues in the next weeks. But let me just show you a couple of examples. So what is also available are the processing pipelines that generate the derivative data from the raw imaging data that are needed to construct these brain network models. And there are two publications that list the software. This was our first publication on this matter, and now the more advanced pipelines that have been further developed since 2015, the first publication, are available in form of containers. And this means that they can be pulled to any system and executed system independently. So you can run all the pipelines on your own machines independent of the system that you have deployed, and also on supercomputers or high-performance computers. And the pipelines provide all the processing steps that are required to achieve the ingredients for our brain network models. For example, the structural connectomes, the parcellation. There are many different parcellations that you can choose from to then construct the model, run the simulations, and you see different types of visualizations of our simulations. This is a glassar parcellation, for example, and the projection on the surface. This is an EG simulation projected on the scalp surface. This all is doable with the virtual brain. We have forward models taking into account the different layers, the physics of the different layers of the brain and the skull to simulate EG activity based on the neuronal activity. There are interfaces that you will also see during the presentations in the next weeks to incorporate information from other digital atlases that are also available on eBrains that follow certain standards in terms of the parcellations, the coordinate reference systems, and therefore they are easy to integrate on our models. And with these automated reproducible pipelines, we can process large amounts of brain data, for example, UK Biobank data or cohort data. There are also special pipelines, for example, for stroke brains, for lesion brains that take into account the challenges that are provided by these brains that differ from normal adult human brains. And you will learn the details also, for example, in the session provided by Patrick Bay. So this is just a little preview of the recently developed processing pipeline for stroke brains that is also available, fully containerized. We just published the preprint for the study. And we also are in the process of sharing in GDPR compliance the processed data sets, in this case, 36 stroke brains coming from our Hamburg collaborators at 4 different time points and 15 controls. So this data will be accessible not publicly, because this is not possible given the biometric information in the data, but following the requirements of the general data protection regulations, which we also will detail during the fall course. Another example of what is available, I have provided to you already. So directly in the cloud, you can run simulations using all the graphical user interfaces of the virtual brain software. Then we have developed the multi-scale simulations that I mentioned before, where we can couple different simulators with different degrees of resolution to also simulate some regions at a finer resolution. You will see a use case that will be presented by Jill Meyer, where we use multi-scale simulation for deep brain stimulation. You will also see, especially I think in the presentation by Leon Martin, how we use information from digital atlases like receptor distributions, for example, to inform our virtual brain models. So here you see the receptor types, and we have the distribution values available from the Jülich atlas, which are now here visualized with different colors that can be directly integrated in the simulations. And here you see another merging of a brain based on MRI data with some tracts derived from diffusion tensor imaging data with an electrode inserted in the basal ganglia. But we see also that the avatar is enriched with cytological information, with microscopically detailed data coming from the Jülich atlas, with data stemming from deceased dead patients, which also can be helpful in informing our brain models. So the goal is really, depending on the use case and the research question, to bridge the different scales and to incorporate the information that is necessary for the specific question. And this includes bridging many spatial and temporal scales and going down to the molecular scale. So here we have an illustration of pathways that include molecular cascades or pathways that often are altered in brain diseases. And obviously the molecular processes are at a very small spatial resolution, can span different temporal scales. But the goal is to also take into account in our simulations, if necessary, even altered molecular processes. And we will show you how this is feasible, so that at the end, we get an understanding if there's an altered molecular process, how this affects the large scale dynamics that we measure with EEG and with MRI, fMRI, or PET. And this is another illustration how we use the information from pathway inventories. So, for example, our collaborators at Fraunhofer Sky Institute developed such pathway inventories that derive their knowledge from literature mining, automated literature mining, and that provides information how certain cascades of interactions, including on the molecular, proteomic, genetic scales, are altered during certain diseases, such as Alzheimer's disease or certain forms of epilepsy. And this information can be linked to some parameters and variables of our brain network models and inform our models and we can investigate systematically how this changes the dynamics. This will be demonstrated during the course. Another possibility is to link the virtual brain to neurorobotic simulators. As we can see here, there's actually a pilot study or proof of principle that has been prepared by Dionysius Perdiccas, who will also give a course session here, and I will encourage him that he also presents this interface, which I think is quite appealing and highly relevant for clinical applications, because it might improve the control, the BCI control of exoskeletons in stroke rehabilitation processes, for example. And this is so you see here collaborations with two startups, Eodyne and BitBrain, who are directly transferring the insights that we generate with our technology development and our related research into applications for patients. There are actually many clinical studies using the virtual brain, and the overview here is absolutely not complete. There's a clinical trial ongoing in the field of epilepsy. There's research ongoing in the field of brain cancer, genetic variants, and psychiatric disorders, stroke, dementia, traumatic brain injury, and many more. And some of these studies we will detail during the course. There are also several innovations. So the beginning was really the brain network modeling approach, where we have this structural skeleton, usually derived from the fusion tensor imaging data and tractography, and then the mass models that sit in the regions and are interacting. But since this, we have many innovative novel developments, for example, multi-scale co-simulation, the possibility to investigate drugs digitally using the virtual brain, in silico deep brain stimulation, the inclusion of multi-scale knowledge in our models, the embodied virtual brain with the link to neurorobotics devices, and the cognitive virtual brain, where we now really start being able to capture cognitive function and even predict with our models cognitive behavior of individuals. Here I just give you a glimpse in a study that probably will be demonstrated in more detail by Leon Stefanowski in the session on neurodegenerative diseases. But here we incorporate PET data, positron emission tomography data, that capture the load of beta amyloid in our brain network models. We couple the load of beta amyloid via this transfer function to the coupling of the inhibitory neurons and the excitatory neurons. So we determine the degree of coupling between these populations with this transfer function as a function of the amyloid beta load that is derived from the PET data. And by just including this additional information, we were able to show that we can simulate or capture, reproduce the slowing of the EEG that we find in Alzheimer's disease patients that have an increased concentration of beta amyloid. And this is visible here. So here you see the simulated EEG of an Alzheimer's disease patient with very slow oscillations compared to the simulation of a healthy control subject, where we have the normal alpha activity in the range of 10 Hertz. This is also visible here, where we have an increase for the Alzheimer's disease digital brain twin in the theta range, where usually we don't have a lot of activity in healthy humans. So the focus with the virtual brain is on understanding mechanisms, how different entities are mechanistically linked to each other, and understanding the principles and rules. And this is just a demonstration how this is possible based on the use case of the previous slide. So what we show here is the state space spent by the activity of pyramidal cells and inhibitory interneurons of a single node, a single brain region, as a function of the activity of the network, the brain network in which this population is embedded. And depending on the activity, the pyramidal cells and inhibitory interneurons can have different dynamical regimes, yeah? And they can qualitatively change, and these points where there's a qualitative change of the dynamic repertoire are called the critical points. And we can now look how this dynamic behavior changes, not just as a function of the embedding network activity, but also as a function of the coupling between the pyramidal cells and the inhibitory interneurons. That is dependent on the load of the beta amyloid, as we have seen in the previous slide. So now we change systematically the coupling between the pyramidal cells and the inhibitory interneurons. This is the parameter J, the control parameter that is now stepwise changed. So you will see that it changes over time. And with every change, we will see that the dynamic repertoire of our simulation also changes. So the geometry of this object is changing, the limit cycles, yeah, and the trajectories of activities on the surface of these limit cycles. So now we can translate these trajectories in the state space directly into time series of local field potential. So this is just another form of visualization of activity in a state space. And we see now, let's wait a second. So for example, this yellow trajectory, if we display it on a time axis, we see an oscillatory signal. And we can now understand why, when we change this control parameter J, the coupling between the pyramidal cells and the inhibitory interneurons as a function of the protein beta amyloid that is increasing in disease patients, why we see a slowing of this electric activity, the local field potential activity that then sums up to the EEG, the electroencephalogram. So this shows you how we really can understand now the mechanisms. So how one change of one entity in the brain, be it, for example, the concentration of beta amyloid, that leads to change of the coupling between inhibitory interneurons and pyramidal cells, leads to change of the large-scale observations, the EEG, and also the fMRI, as you will see later. And these simulations can be also used as mathematical or computational microscopes. So by constructing such models, brain network models that reproduce, for example, resting state fMRI activity or EEG activity, we at the same time receive all the state variables of the neural mass models that we have used. And the state variables have information, for example, on the firing rates of the population models that we use. This is information that is usually not achievable, not measurable non-invasively in these subjects or patients or persons. So this is additional information that can be used to enrich the feature space that is spent by the empirical features. And the details will be also presented by Leon, can be used to augment machine learning-based classification of dementia. And we have shown here in the study by Leon Stefanowski that indeed we can significantly improve the classification if we run first such simulations that enable us to come closer to the actual neuronal processes. And this is a quick example on virtual drug therapy, probably also detailed by Leon, where we can now simulate the effects of drugs, yeah, on the computer based on our avatars. So what we did here, we simulated the effect of an NMDA antagonist, memantine, that is used to relieve the symptoms of dementia. We have here the power spectrum of different individuals, Alzheimer's disease individuals, mild cognitive impairment individuals, and healthy controls. And this is a simulated power spectrum, so it's not the measured empirical data. And we see the slowing in our simulations of the EEG activity of Alzheimer's disease patients. That is a consequence of the PET data and the high beta amyloid load that we included in our simulations. So now if we include a mechanism, an NMDA antagonistic mechanism in our model, which mimics memantine, we can see that we can reverse the slowing in the EEG in our simulations. So we can mimic the effect of this drug, but we can not only reproduce the effect, but we get additional information, and this is visible here. But first of all, this reversal or decrease of theta activity of the slow activity upon administration of the NMDA antagonist memantine has also been shown empirically. So what we show here reflects the reality. And now if we look here in this diagram where we display the output activity of the pyramidal neurons as a function of the memantine, the virtual memantine application, the concentration of the memantine. And here we see on the x-axis the activity that drives the pyramidal cells, that goes in the pyramidal cells. And with increasing activity that drives the pyramidal cells, there's also an increasing output of the pyramidal cells naturally. The problem is that Alzheimer's disease, it's postulated that the inhibitory interneurons fail. And therefore, there's an increase of excitatory activity and excitatory output in the pyramidal neurons that leads to excitotoxicity and cell death. As we can see here now, if we increase the concentration of memantine in our simulation, so this is a simulation here, the input-dependent increase of the output of the pyramidal neurons is buffered. Yeah, so here you see that with increasing input, there's an increase of output activity, but here with a higher dose of memantine, we don't have such a relationship. And this may be the protective mechanism of memantine, which is nicely illustrated here in a mechanistic way. So I mentioned the possibility to use multi-scale simulation for deep brain stimulation, and these are just a couple of additional illustrations and first insights into the results that will be presented in much more detail by Jill Meyer in the dedicated session, where you also probably have the possibility to work on the code, to get access to the code and to the data to reproduce these simulations. But in a nutshell, what is now possible with the multi-scale simulations is that for the regions of the brain, in this case the basal ganglia, that are captured at a fine degree of detail, at the degree of spiking neuron networks, we can generate actually not only the spike rates, but the spike times. Yeah, we really get the spike timing of the individual nodes, as you can see here. These are the basal ganglia and the thalamus. And with this, we have the possibility to include, for example, spike timing dependent plasticity rules and enable plasticity. So the connectivity in the network changes over time as a function of the activity. But at the same time, while we are simulating the basal ganglia at a resolution of single neurons, we can monitor the activity in the remaining brain, as you can see here. And this is highly relevant also to predict the outcome for the patient and possible side effects. And here you see a demonstration of a thalamic activation upon stimulation of different brain regions or basal ganglia regions. So this is the off stimulation scenario. And here you see for stimulation of the globus pallidus internus, the subthalamic nucleus. This is a biphasic stimulation, and the subthalamic nucleus with a monophasic stimulation, the increase of the firing rate in the thalamus. And this is actually what is desirable for the patient, but what is also desirable is to understand what are the effects in other regions of the brain. And for the same 3 stimulation scenarios, we see here the cortical effects, and we see that while in the thalamus, the effect is pretty similar, we have quite differential effects in the cortical regions, including a deactivation of motor cortices upon monophasic stimulation of the subthalamic nucleus compared to biphasic stimulation of the same area. Okay, now we take a little rest, and before we have a short break, I just want to show you how we use the virtual brain also for public outreach activities. And what we have developed is a software that informs about the brain. That is our interactive brain atlas that is available in many languages, and that is made for the broad public. I put the link to this application in the chat again, so you can go there and try it out yourself. It takes a little bit to load. Be patient, please. It takes maybe 30 seconds or so, maybe less. But as you can see here, you can then scroll through the brain regions. You can also select functional networks, and you get information about the regions and the functional networks in many different languages. And this is the basis for applications that we prepare. This is just another language, obviously, that we prepare for public outreach activities. So some of the applications run on mobile devices. They also can connect to wireless EEG headsets, can be used for neurofeedback games. But they are also used, for example, for exhibitions. This is an ongoing exhibition here in Berlin in the Humboldt Forum, where, for example, there are such virtual brain visualizations in the Central Media Tower. But there is also space somewhere here, I think, which is called the Humboldt Lab, where there are interactive stations where naive individuals can learn how to set up a brain simulation and what the different measures of brain activity actually mean. So if you're in Berlin, I only can invite you to go there and visit and explore this exhibition. There's another ongoing exhibition in the Museum for Medical History at the Charite Berlin, which is relatively new. It opened just, I think, a couple of weeks ago. I think at the beginning of the summer, where you also have the possibility to interact with the virtual brain, but there are many other things you can do. And there's an exhibition in the Museum for Communication in Berlin where we actually developed a special version of our interactive atlas on the topic of argument. They have an exhibition on the topic argument. And the curators asked us to visualize networks that are related to emotions during arguments. So there's a tool actually that has been developed by the team that will be also presented by Leon Stefanowski and Leon Martin, for example, where we can, from the literature, through literature mining, obtain networks that are associated with certain concepts, like, for example, emotions that are related to the topic argument. And we did that. And these networks that have been identified, for example, related to conflict, to controversy, to debate, can be selected by the audience and visualized, and some additional information is made available. So this would be also a possibility for you to explore. The Museum for Communication is really nice. And if you happen to be in Berlin, just go there. The exhibition is ongoing. I think it lasts 1 year. Then it will move further to Frankfurt. And there you can find this interactive atlas. And with this, I would say that we take a little break now and we continue with 2 topics for the second part. The first topic is GDPR. This is something you need to know. I hope you can see my slides. If not, please let me know. And the second is probably more exciting. It's a demonstration of two use cases where we learn to understand true mechanisms related to brain function. And this shall demonstrate to you the power and the potential of TVB applications. But we start with a maybe, for some of you, more boring part, but equally relevant part, the general data protection regulation. So the data that we are using here and the digital twins that we are producing are highly sensitive. They contain health information and personal information of human subjects, which is highly protected in the European Union. And there's a law related to that, which is called the General Data Protection Regulations. And therefore, all the processing of such type of data needs to be done in compliance with GDPR, else the processor or so-called controller. So if you determine the purpose of the processing, if you are doing a research project, for example, then you automatically have the role of a data controller if personal data are involved. And with this, you are responsible and liable. And the data subjects could sue you if you are not properly protecting the data. So therefore, a big effort of our team is on developing infrastructure to enable the research on digital human twins. This is just to give you an overview on the investment in this topic. So I am myself the coordinator of this EOS project that just ended, that has been funded with 15 million euros over the last 4.5 years. Then since last year, I'm coordinating the eBrain Health Actionable Multi-Level Health Data Project funded with 13 million euros, lasting 4 years. And just in this year, this big project started funded with 60 million euros, which is called Testing the Experimentation Facility for Health, AI, and Robotics. And the same topic of all these 3 projects is to develop infrastructure for data-driven, health data-driven innovation. So this is a big topic, and it's not an easy goal given the regulations in the EU. But we were successful in developing such infrastructure, and it's actually accessible now also via the eBrain's website. Unfortunately, I don't have now here the link, but maybe one of the colleagues can put it in the chat for you. So if you go to the eBrain's URL and you go to health research platforms, it's a little bit hidden, and then you go down to work with health data, you will get access to the virtual research environment and the health data cloud, both developments under lead of the Charite that produce such infrastructure for sensitive data. The virtual research environment is fully functional and operational and audited and provides such a space for working on digital human twins. It's also fully, the health data cloud is basically a further development of the virtual research environment towards the federated infrastructure in Europe with several nodes, and it's fully integrated with the eBrain's infrastructure. So again, we have a single sign-in. If you go here in the test bed of the health data cloud itself is not yet audited. Therefore, we call it a test bed. We didn't go through GDPR certification yet there, in contrast to the virtual research environment, which is fully certified. So this is a virtual research environment, and just to give you an idea, it can be accessed via several registries and marketplaces, for example, the EOS marketplace or the DFG resources, the German National Research Foundation resources website. It provides access to data sources in the Charité. So for example, the radiologic imaging centers in the clinics and for research are connected with virtual research environments, a health data platform that collects all the clinical data And high-performance computing, which is also GDPR audited, is directly connected to this virtual research environment. There's a portal where researchers also from outside the Charité can get connected with the VRE and the functional features provided by the VRE. And then there's the actual function. And this is a wide functional scope. We will not have time to go on the details. There will be a session, I think it's the last session of this course, where we will have a demonstration, but we also have related weekly community meetings. Unfortunately, I don't have the link here, so I can give it to you later after I stop the screen sharing, where you can register. It's open for everyone. And this week actually is very interesting. The community meetings take place every 2 weeks on Friday at 4 p.m. And this week we have a very nice comprehensive demonstration of the virtual research environment with the entire functional scope, a live demonstration. So if you are interested in this environment, I invite you to register. As said, I will provide the link later and participate in this demonstration on this Friday. This is, as I mentioned before, now further developed in a federated, decentralized architecture across Europe with the participation of several data centers. This is probably a little bit difficult to understand graphic, but the message is there's a central node, there are satellite nodes at Charité, for example. This is eBrains, several other data centers. Several of them are already, as the Charité data center, a critical infrastructure with high level of information security, and they're interoperable, and there's a central knowledge graph. So the data that are located here at the Charité can be discovered via the eBrains portal, the eBrains knowledge graph, by the global user community. And the data are safe here at the Charité. They don't need to be moved to eBrains or to the supercomputing centers that power eBrains, but they are also accessible. So upon concluding of all the necessary agreements, researchers who have discovered the data via the central knowledge graph get access to the VRE and can process the data securely in the virtual research environment at the Charité or in other data centers at Oslo University Hospital, for example. And this is the knowledge graph of eBrains. You can find it via the eBrains portal. Maybe one of the colleagues also gives you the direct link. There, it's actually, I think, an older screenshot. The number of data sets now is a bit larger. There you have several data sets on a European scale that are being made discoverable via this graphical user interface with keyword search, but also programmatically. You can not only search for data sets, but also for computational models. There are many, as you can see here, and software and tools. So everything is annotated. There are metadata models, not only for data sets, but also for tools and software and computational models. The metadata schemas are being developed by the community and are evolving. So they're getting more detailed depending on the use cases. And it's exactly this metadata, the annotation of the data and the tools, that makes the data and tools discoverable. So it's a very important component. And we are developing ourselves tools to generate such metadata for our data. So when we are processing, for example, raw data that comes with metadata, we bring the metadata in the appropriate format, a standardized format, and then the metadata are actually connected even to the derivative data sets. So each derivative data set will be attached to the metadata to make sure that future users of the derivative data have all the information available that is required to make full use of the data. And this is just an example of one of the studies that I will talk about later that uses the Human Connectome Project data to construct 650 individual virtual brains to understand principles of cognitive function and the human brain. The data come from the US, but as soon as we are processing them here in the European Union, we have to follow European Union rules. They are sensitive data, they contain information about health, and therefore they need the right level of protection, and therefore they are stored now securely in the virtual research environment at the Charite. But you will be able to discover them, the non-sensitive metadata of this data set on eBrains. So if you, for example, actually, since we just provided the data sets, I think it's on top already visible, but you can also search it by putting in, for example, the names of the authors, Michael Schörner, or my name, or Human Connectome Project, you will find them. And importantly, the dataset card that you will receive with each dataset already contains important information, so you can make sense of the data, but also information about the license and the options how to access the data. And in the case of sensitive data, this includes not only license information, but also information about the contractual agreements that need to be concluded before you can access the data. And if you explore this further, you will see that you are guided to a so-called collaboratory where all this information, all the contracts that need to be signed and filled first, this information from the secondary data user are available. And there we use another resource of eBrains, the so-called collaboratory. These are collaborative workspaces where you can store information. You can keep the workspaces private or make them public or invite collaborators. This is another another functionality that is provided by this digital infrastructure, eBrains, which is quite useful and which we will use in the course of this fall school. So I think so far I have shown you that you can use the virtual brain for clinical applications to improve the diagnostics or the prediction of disease trajectories, to understand mechanisms, and even to run virtual therapies. But now I want to dive a little bit deeper into some projects done with the virtual brain that really reveal mechanisms. And one of the projects that shows this nicely is not presented by me today. It will be presented by Dominic Koller in one of the later sessions on the generation of traveling waves. But today I will touch two studies of our lab. First of all, this eLife study, inferring multi-scale neural mechanisms with brain network modeling. And then second, this study recently published in Nature Communications, learning how network structure shapes decision-making for bio-inspired computing. Let me just look at the time. We have not so much time left, so I will try to make it as concise as possible. So what we see here is obviously virtual brain with a simulation of resting state activity. And in this eLife study, we digitalized the brains of 14, I think, healthy adult individuals and simulated 22 minutes of resting state fMRI activity. And we used the reduced Wang-Wang model. These are the equations that describe the state variables of the populations. We see the input currents that drives excitatory population, the input currents that drives inhibitory population, that translate then to the firing rates of the excitatory population and the inhibitory population, that translate to synaptic activities of the excitatory and inhibitory populations. And while we are now simulating with this brain network model, the resting state bold fMRI activity of each individual, our underlying neural population models generate in each brain region exactly these state variables, including the firing rates, for example, that are shown here for one example region, firing rates of the excitatory populations and the inhibitory populations of this reduced Wang-Wang model. And we now can investigate the behavior of these state variables and relate it to other experimental observations. Yeah? And this is what we have done here in this overview. where we actually sorted the different features according to the time scale, starting with a very short time scale of 100 milliseconds. And here on the right side, we have the longest time scale of 20 minutes or 22 minutes. In the upper panel, you see the simulations, the analyzed simulated signals. On the lower panel, you see the experimental data coming from different studies, even from different species, monkey, rat, monkey, and humans. And we now look at the behavior of the state variables of the model that I have shown you in the previous slide, and look at the different relations and how they compare to what is known from empirical data. So here, for example, we look at the firing rates in relation to the phase of a single alpha oscillation. And we see that here at the peak of the alpha oscillation, we have the trough of the firing, the minimum of the firing rate. And a similar relationship can be found in monkey recordings so invasive recordings of the firing rate and the local free potential. So then we move in the second panel, where we relate the amplitudes of inhibitory postsynaptic currents and excitatory postsynaptic currents in our simulated signal, and compare this to what has been measured in rats. And we see that in our simulations, the amplitudes of the inhibitory postsynaptic currents are multiple times, about 5 times larger than the amplitudes of the excitatory postsynaptic currents, which compares well to what is known from empirical recordings. Next panel, we compare the firing rates, this time not with the phase, but with the power of the algorithm. And we see that with increasing firing rates, we have lower alpha power, or the other way around. With increasing alpha power, we see a decrease of the firing rate. And this is what is also found in invasive recordings in the monkey. We look at the relation of our simulated EEG alpha power to the simulated Bolt signal in our simulations, and we see it's inversely related, it's anti-correlated. This has been shown in simultaneous EEG fMRI recordings in humans many times before. We look at the power law behavior of resting state fMRI in our simulations, and we see scale-free behavior as we can find in empirical data. And last but not least, we can look at the switching of the functional connectivity over time, and this very well compares to what can be seen in resting state fMRI data. So now, in contrast to the lower panel, where we have snippets taken from empirical data that are unrelated, all the observations in the upper panel are mechanistically linked. They are the outcome of a single mechanistic model. And we now can understand how these observations are mechanistically related. So if we would change, for example, the ratio of inhibition excitation, how would that influence all the other observations? This is the beauty of these brain network models, that we can now link the many different observations, the knowledge that we have obtained from empirical data in a single theory, in a single unified computational model. And sometimes things will probably not fit and we have to make adjustments, but this is how iteratively we learn and to understand how the neuronal interactions in the brain emerge. And I want to demonstrate this in more detail with these analyses, where we look at the actual state variables. So you might recognize the terms. I have shown you the equations in a previous slide. We see here the long-range input currents. We see the synaptic input currents, the synaptic activity, the firing rates, and the synaptic gating. These are all state variables of the model. And we see here the symbolized excitatory and inhibitory population of the brain region. The state variables come from our model, and they have been averaged here according to the zero crossing of a single alpha cycle, which you see here. So what you see here are the averages, the grand averages of millions of trials of our state variables, yeah, and there are no error bars because they are so small. So these are grand averages of the state variables averaged according to the zero crossing of a single alpha cycle. These are the input currents that drive the excitatory and inhibitory populations, and they summed up. This is the global excitatory inputs, this is the local excitatory inputs, excitatory postsynaptic currents, and the local feedback inhibitions, inhibitory postsynaptic currents. The scaling of the input currents is different for the excitatory population and the inhibitory population. It's indicated by the right axis for the inhibitory population and the left axis for the excitatory population. So now we can sum up the large-scale network inputs, the local excitatory inputs, and the feedback inhibition. You remember the large-scale equation of TVB. Yeah? These are exactly the components shown there. They are weighted according to these scales. These are summed up, yeah, for the inhibitory populations, just the local excitatory postsynaptic currents and the feedback inhibition. This is a summed up activity. It drives the inhibitory population. This is the output of the inhibitory population in terms of the firing rate, the synaptic activity. The synaptic activity of the inhibitory population has an inverted effect because it is an inhibitory. Therefore, the curve is inverted. And we see now that actually it is also inverted in relation to this alpha cycle. And we sum up this feedback inhibition now with a local excitation and with a large scale input from the network. If we look at the axis that scale the activity that drives the excitatory population, and we see that the inhibitory feedback signal dominates the summed up activity that drives the excitatory population. Therefore, the shape of this input is very similar to the feedback inhibition signals that you can see here. And this leads to the following firing rate and synaptic activity. So we see that because of the feedback inhibition, the input that goes in the excitatory population and that leads to firing rate is inverted compared to this alpha cycle. So we see that at the peak of the alpha cycle here, we have a drop of the firing rate. There's a pulsed inhibition, a phase-dependent pulsed inhibition of the firing rate in our model because of the ongoing excitation and inhibition loop here. And this also leads to pulsed synaptic, to pulsed inhibition of the synaptic gating variable. So here we see the mechanism of phase-dependent pulsed inhibition of the excitatory firing rate on the scale of a single alpha cycle that lasts 100 milliseconds. Now we can zoom out, but before we do this, we compare what we see in our simulations. So this again, we have seen it in the previous slide, is the relationship of the firing rate and the alpha phase in our simulations. This is what has been observed in the Marquet. So our simulation nicely, nicely is aligned with the empirical observations. It's also aligned with other empirical observations. So here you see the traces of inhibitory postsynaptic potentials and excitatory postsynaptic potentials. And we see that if there's excitation, it is closely paralleled by inhibition. This is exactly how it happens in our model, if you remember. We look now here at the amplitudes in the pyramidal cells of inhibitory postsynaptic currents and excitatory postsynaptic currents, and we see the amplitude difference that we have also in our model. Inhibitory amplitudes are 5 times larger than excitatory ones. And what we also have in our model and see in real empirical data is a little time delay between the excitation and the inhibition of a few milliseconds. Yeah, this is also in our model. So our model nicely captures all the different observations on the empirical side. So now we zoom out. We do not look on a time scale of 100 milliseconds of a single alpha cycle, but an entire burst of alpha oscillations with a power increase, alpha power increase. So the alpha power is the power of the 10 Hertz oscillations here, indicated by the orange envelope. And we look now what happens when there's such an alpha power increase. It's the same thing. We have again the state variables, averaged, grand average, and we follow them through the excitatory and inhibitory population. We start with inhibitory population, where the sum of the local excitatory postsynaptic currents and inhibitory postsynaptic currents goes in. We see here that we have a rectification of the firing rate. Yeah? Here we have this spindle activity around the zero line. And here's a rectification because there are no negative firing rates. And then again, we have this inversion because of the inhibitory effect of the inhibitory population. And now we sum up the large scale network input, the local excitatory signal and the feedback inhibition. Again, there's a dominance of the feedback inhibition that drives excitatory population. Therefore, the summed up activity is very much dominated by this feedback inhibition activity. And this leads to a pulsed inhibition again of the firing rate and the synaptic activity, and consequently the fMRI signal. So if there's an increase of the alpha power, then we see a drop of the firing rate and of synaptic activity in fMRI. And this is exactly what we find in empirical data. And here we see the mechanistic, the postulated underlying mechanisms. So again, if we compare simulated and empirical data, we here look at the relation of the firing rate and the alpha power in the simulation and in real empirical data in the monkey. And it's very similar. And yeah, I don't have time to look in the simultaneous EEG-fMRI, but I have shown that before. So let's then zoom out another step. So now we do not have a single increase of the alpha power, but multiple increases. So we have a certain cycle frequency that modulates the power. We have slower modulations and faster modulations of the alpha power, the cycle frequencies of 0.1 and 0.3. And again, we follow through the local excitatory and inhibitory population. We sum up the excitatory postsynaptic currents, inhibitory postsynaptic currents. We see again, we have a rectification, we have an inversion. We sum up these ingredients that are dominated by the inhibitory feedback, which leads to a alpha power dependent pulsed inhibition of the firing rates, synaptic activities, and the fMRI signal, exactly as we see it in empirical data, but here we have it in an integrated theoretical model. What we also observe is that depending on the cycle frequency of the power modulation, whether it's slow or fast, we have here different amplitudes in the firing rates and synaptic gating activity and fMRI. So there's a translation of the cycle frequency in the amplitude. And if we look closer, we see that this relationship emerges in the global network activity. So it's a long-range excitatory input that shows here this relationship. Yeah, and we also know why that is, because there's a pretty slow time constant of the excitatory transmission of, I think, 100 milliseconds. And therefore, if there's longer increase of alpha power, there's an accumulation in the network that leads to higher amplitudes. If there's less time, there's less accumulation of excitatory activity. But this is the basis of the scale-free behavior, the frequency-amplitude relationship that we see in resting state data. And it's also a consequence of the network, because if we now turn off the network here and only look at a single population model, we see that this relationship between the cycle frequency and the amplitude of the firing rate and the fMRI signal disappears. So we need the network, the complexity of the network, in order to have this scale-free behavior in our resting state data. And this is also shown here. So we see here the power law behavior of empirical fMRI data with the exponent, the beta exponent that determines the steepness of this power law behavior. We see very similar behavior in our simulated brain network model. But if we turn off the global network and only look at a population model of the single unconnected node, the beta exponent changes and is very different, very flat from what we see in empirical data. So this is just a summary slide showing the many different parts together And yeah, I hope I could provide you a demonstration how we really, with this approach with brain network modeling, can understand at different scales how the processes are mechanistically linked. And so this is just a summary. Ongoing EI balance in a brain network model leads to fast pulsed inhibition, slow resting state network fluctuations, and power law behavior. And the model explains how neuronal units interact and give rise to several observations and their causal relationships. And now I come to the last part of my presentation. I just check the time. I think it should be still sufficient. The very new study that recently has been published in Nature Communications, where for the first time we look really in cognition with the virtual brain. And this is just a visualization of some of our analysis. So what you will see is that we develop brain network models based on 650 individual brain imaging data sets. And we plug in these individualized brain network models functional circuit models that have been shown previously in an isolated way to be able to mimic decision-making and working memory. But now they are embedded here in the individualized brain network models, and we investigate if they perform differently, if the decision-making differs depending on whose brain avatar we are using. And indeed, it differs, and it is tightly correlated to the actual functional behavior of the respective individual. So first of all, in the study, we did further development of our approach, and this is very important. We introduced, in addition to long-range excitation between the excitatory populations of different brain regions, feed-forward inhibition. And this is something new. This was not done before. So we now can regulate the relation between long-range excitation and feed-forward inhibition. And the feed-forward long-range inhibition does exist in the biological brain. Yeah, it's well known, well described, but it has been ignored in the past in these brain network modeling approaches. So here you see an example where we have a high long-range excitation and a low feed-forward inhibition indicated by the thickness of the arrows. And here it's the other way around. We have a low long-range excitation and strong feed-forward inhibition. And this is now our tool to regulate synchronicity in our brain network model. So it has been shown that by just modulating the ratio of excitation, long-range excitation and feed-forward inhibition, we can regulate the degree of synchrony or anti-correlation, correlation, and no correlation between brain regions, as you can see here. So this is a simulated signal. This is a functional connectivity, and here you see the excitation inhibition ratio. And when we change that, we go from anti-correlation to no correlation to high correlation. So we have now a nice control mechanism to adapt to just our brain network model to what we find in the actual functional data of our individuals. And this is a tuning process using this mechanism. We see that now it's possible by tuning long-range excitation feed-forward inhibition to really closely reproduce the empirical functional connectivity of an individual with a correlation of close to 1. So it's an almost perfect reproduction of the functional connectivity of an individual. And with this, we now have the possibility to understand in a much better way how this functional connectivity topology or organization affects cognitive processing in our model. And so this is what we have done. As mentioned before, we use already evaluated circuit models that have been shown to be able to mimic decision-making and working memory and plug them in these optimized individual brain network models in the prefrontal cortex and the parietal cortex. And we now, as has been done before, can derive this circuit model where these two populations are sitting in the prefrontal cortex and these in the postparietal cortex, and they are competing. We derive these populations with evidence. Yeah, so these are input currents. Yeah, you see here the unit ampere, and there's a certain contrast. Yeah, you can see that the evidence here is a little bit higher for population A than population B. And this means that the correct decision would be for population A because the evidence is higher. But the challenge is that there's noise on top of the evidence. And this makes the circuit model fail, depending on the degree of contrast. If there's a very high contrast, it's easy. But if there's a small contrast, as you can see here, and a lot of high amplitude noise on top, then the circuit model can fail. And despite stronger evidence going in population A, population B might be the winner, and the decision might be for population B. And how this looks like in the simulation, I show you here. So this is a scenario where the decision is correct. These are the time courses of the simulated neuronal activity for all the 4 populations. You see the color codes. The blue traces are for the populations A, the green traces for the competing population B. And the evidence is stronger for population A, and indeed population A here wins and reaches the high attractor state, which leads to the decision, the correct decision, one correct decision. And we can run now the simulation again, get another correct decision. Let's see what happens now. Now we have another scenario, an incorrect decision, because despite the evidence is higher for population A, the population B gets in the high attractor state, and the decision is taken for B, which is a wrong decision. And this we can do now for our 650 individualized brains 1,000 times each. Yeah? So we let them take decisions. The circuit model is embedded in the individual brain network models. And we get now a certain ratio for each individual of correct decisions and incorrect decisions. And in this case, we have 790 correct decisions and 210 incorrect decisions. The interesting insight is that this ratio changes for each individual digital avatar. And we compared the performance to the actual performance of the individuals to which the avatars belonged. And we could find that the brain models of smarter subjects with a higher fluid intelligence, a better capability of solving difficult problems, made more correct decisions and took more time for decision making, like the actual, so exactly like the actual subjects. This is shown here. So there's a correlation between the fluid intelligence score of the individuals and the simulated decision-making performance. But we also found that there's a positive correlation between the simulated decision-making time and the score, the fluid intelligence score. So this means that the people, the individuals with a higher fluid intelligence took longer to come to a decision in our simulations. So what is the task that the actual individuals had to do? They had to solve the Ravens-Pence progressive matrices, the PMAT task, which consists of 24 individual tasks that require the subjects to complete these patterns. So they have to find the logical rule that leads to the final pattern. And the difficulty of these tasks increases with each task. So the first task is very easy. The last of the 24 tasks is the most difficult one. So if we now look at the performance of the real subjects, not of the digital avatars, we see here we have the G factor of intelligence, the general factor of intelligence. For the first very easy task, and we see that the reaction time is negatively correlated. So the higher the fluid intelligence, the faster they reacted for the easy task. for the most difficult task of this PMAT test, we see the relation inverts. So the more intelligent individuals had longer reaction times. And we can look at this then on an individual task level, on group averages and individual subject level. So what we see here in yellow are the correlations between the fluid intelligence, the PMAT score, and the reaction time. And in brown, we see the correlations between the general factor of intelligence score of these individuals and the reaction time. And we see that for the easy tasks, there's this inverted relationship. So the people with higher fluid intelligence or general factor of intelligence react faster. But then from a certain level of difficulty on, this inverts. And the people with a higher general factor of intelligence and the higher fluid intelligence start reacting slower. And this is maintained for the more difficult tasks and can be seen on an individual subject level as well. So we found actually after seeing this in our simulations that really the individuals that have a higher fluid intelligence and general factor of intelligence take more time for solving difficult problems. So now let's go back to our simulations. When we tune the functional connectivity, the degree of synchronicity between the networks, we at the same time also influence the degree of input correlations of the currents in our network, as you can see here. And we also influence the input amplitude, the amount of currents that drive a population. Yeah, so this is what you see here. So by changing the degree of functional connectivity, we at the same time tune the input correlations of the currents that drive a population and the input amplitude. And here we see how in our models, the degree of input correlation is correlated with the functional connectivity, and the input amplitude that drives brain regions is anti-correlated with the functional connectivity. So there's a very clear relationship between functional connectivity and input, neural input correlations and the input amplitudes. You remember the diagrams I have shown from the ELIVE study where we see the input currents. This is exactly what we are talking about here. So now we wanted to see how for different states of synchronicity in the network, our circuit models do perform. So we took 3 scenarios: low network synchronicity, intermediate network synchronicity, and high network synchronicity. And we run the 1000 simulations, and we see that for low network synchronicity, we have 210 incorrect decisions, intermediate 184, and for high synchronicity, we have 41 incorrect decisions. So the best performance we achieve for high synchronicity in our networks. So now, since we have mathematical descriptions of all the processes going on, we can have a state space analysis and understand why that is. So these are the state space diagrams that are spent by the activity of the population B and population A in the postparietal cortex for the 3 different synchronicity scenarios. And we have started here a simulation and see for 1000 simulations trajectories in the state space diagram. Green if the decision goes to population A, and red if the decision goes to population B. And we see that for the different degrees of synchronicity in the network, we have different configurations of the state space diagram. And the different configurations can be seen, for example, in the different fixed points and the flow fields that lead our trajectories. And the effect can be observed. So for the low synchronicity scenario, we see that very quickly the simulations, the trajectories go to either of the 2 fixed points. So they very quickly go towards one decision led by these flow fields. So this is not the case, obviously, for the high synchronicity state, because as you can see here, we have a fixed point in the midline, a balanced state between population B and population A. which means that it's difficult for the system to either move to one of the two decisions or attractors. Instead, time is generated to accumulate further evidence and also average out the noise on top of the evidence. So now if we continue our simulation, we see that after 1 second, all the decisions have been made for the low synchronicity state. Also now the intermediate synchronicity state has reached most of the decisions, but the high synchronicity state still lingers around this fixed point here. And only if we wait for another 0.5 seconds, we see that in the high synchronicity state, the fixed point is resolved and the system can jump to a decision, very well-informed decision, because there was more time to average the evidence and to average out the noise. And therefore, the proportion of correct decisions is better here. So now I think this is basically the last slide, and it's very full, but I still want to explain to you. Again, we have 5 scenarios of synchronicity, and we now focus only on the first column here, which is a very high synchronicity state. Yeah, and the others are just other synchronicity states that are less high. So what we have here are, again, the time courses of the competing populations in blue and in green. And we defined 3 time points here: baseline, the ramping of the, or here actually time point 2 is the ramping of the frontal population of the winning team. And the third time point is the achieved attractor state. And for these 3 time points, we now plot here the, oops, this was not intended. We plot here the state space diagrams again. And we see that in the baseline time point, we have here this fixed point that I have mentioned on the last slide that prevents the system jumping to one of the 2 decisions or attractors. So now when the activity of the frontal population of the winning team is ramping up, at the second time point, we see that this fixed point is being resolved. So there's actually this ramping up of activity in the frontal node that leads to a reconfiguration of this network that releases the fixed point, and only then it is possible to come to decision. So in other words, it's a inhibition by the frontal network that leads to the prevention of a premature jumping to a decision. And I think with this, my presentation is almost ended. This is just a summary slide where we show again the relationships of functional connectivity, the input amplitudes, the resulting proportion of correct decisions, time until decision, reconfiguration of the state space that explains the different performances of the different individual avatars. And I hope this was inspiration enough for you to be motivated to follow us through the future sessions. I would like to thank all the people from our lab who contributed to the work that I have shown. There were actually many more contributors, our collaborators, that are not shown here, but that are equally relevant. And with this, I also thank you for your attention and, of course, the remaining time